Welcome back to Amoeba Classes, my dear students. As we know that we were discussing about mitosis, the cell division, and in the last class we have discussed about cell cycle and the very first phase, that was interphase. In interphase we have already discussed about the G1 phase, G0 phase, S and G2 phase. Okay, so let's see what we will be discussing today in this session. So let's switch to the scope of syllabus and let's see. So today we are going to discuss about the cell division, and in cell division we will be understanding the mitosis and its different stages. So this will be the exclusive topic for this video. So make sure, I think that this video will be a little bit longer. So make sure that you focus on this video very properly, and let's begin. Let's do a very quick revision about what we have discussed in the last class. Okay, so in the last class we have discussed about the interphase, and we have seen that interphase there is G phase, S phase and G2 phase, and from the G1 phase the G0 phase, that is the resting phase, also arises. That happens in the heart cells, the nerve cells and the permanent tissues of the plants. Okay, now we have discussed that in the S phase what happens, there is a duplication of chromosomes takes place. Now as I've already told you, this is the centromere and this is the chromatid. Okay, on the basis of the number of centromere we'll give the number of chromosomes. So right now one centromere, that is one chromosome, and this is one chromatid. Got it? Okay, and when in the S phase this chromosome will divide or duplicate, what is exactly happening? This chromosome is still one, but the chromatids are dividing without the splitting of the centromere. So what's happening over here? This is one chromosome, this is also one chromosome, but this is one chromosome with how many chromatids? 2 chromatids. Okay, so this was the very important part that we have discussed in the S phase. Now let's take a quick look what exactly is happening inside the animal cell. I will be discussing about the animal cell and later we will be comparing it with the plant cell. Okay, so this is the cell membrane and this is the nucleus, and we know that this nuclear membrane is double layer. Okay, so I'm not making double layer right now, and this is the nucleolus, and this is what, I hope you remember, this thread like structure, this is what, chromatin fibers. Okay, now in the S phase, that is the phase of the interphase, in the S what is happening, that one chromosome, it is converting into 2 chromatids. Okay, and this is what the replication is going on in the S phase. Now over here this chromatin fiber, it is in the thread like structure. Now what will happen after this interphase? Now comes the real phase, that is called as the M phase, and this is what we have to discuss today in detail. Okay, so let's begin. Now this M phase, it is called as mitosis or mitotic phase, and this mitotic phase is divided into 2 categories. The very first category is karyokinesis. Okay, what it is? Karyokinesis. And the next category is cytokinesis. Okay, let's try to understand the word. So let's break down the word. Carry means what? Nucleus. Okay, not that YouTuber Carrie Menati, don't get confused. Okay, anyhow, nice joke it was, I know. So carry means what? Nucleus. Okay, and in class 9th also, if you remember, prokaryotic cell and eukaryotic cells, in there only you might have remember the carry means what? Nucleus. So karyokinesis means division of nucleus. Okay, simple, from the term only you understood that karyokinesis means division of the nucleus, and I hope now you understand what cytokinesis means. Cyto means what over here? Cytoplasm. That means division of the cytoplasm. That means this M phase, first what will happen? Karyokinesis, then what will happen? Cytokinesis. Now the very important thing is this karyokinesis is divided into 4 different stages, and those stages are P, M, A, T. Okay, so what is the full name? Very first one is the prophase. What it is? Prophase. Then it is metaphase, then anaphase, and then we have telophase. Telo means at the last, terminal, right? In the very first lecture we have studied about the types of chromosome, in which we have studied about telocentric type of chromosome, right? So that means the centromere was at the terminal position. In the same way, the terminal, the last phase of the karyokinesis is termed as telophase. And what are the initials we are getting? PMAT. So you can remember this trick: Prime Minister at Amoeba Classes, right? Because you are watching right now what? Amoeba Classes. So you can learn this simple trick: Prime Minister at, okay, Prime Minister AT will be at, and you have to edit this Amoeba Classes, or whatever you wish to like, you can add, not only Amoeba Classes. Okay, so these are the some basic things. Now what we have to discuss in detail, we have to discuss this whole thing step by step: the prophase, the metaphase, the anaphase, and the telophase. Now these 4 phases I will be explaining in detail right now, and then we will be switching to the book and we'll see the remaining major points. So here I will be discussing right now about the important steps, that what exactly is happening inside the cell in the very easy manner. Then we can switch to the book. Now see, we know that in mitosis, the diploid cell, when divides, it forms 2 daughter cells, and they are also what? Diploid. They are also what? Diploid. If haploid cell is dividing, it will be forming 2 daughter cell and they will be haploid. That means in mitosis, the number of chromosomes remains the same after this cell division. Okay, so what we will be doing, we will be taking the example of the diploid cell. As we know that mitosis takes place where? In the somatic cells, and these somatic cells are diploid cells. Okay, so let's begin. Okay, so this is the cell membrane, and this is the nuclear membrane, and this nuclear membrane is a double layer membrane, and inside it is the nucleolus, and inside the nucleolus there is the chromatin material. Okay, this is the chromatin material. Now this is what the interphase looks like. Now when this cell enter in the M phase, that is the mitotic phase. Okay, mitotic phase, what happens in the mitotic phase? Karyokinesis will start, and in the karyokinesis, the very first phase is the prophase. Okay, don't forget, Prime Minister at Amoeba Classes, PMAT. So the very first phase is what? The prophase. Now let's see what will happen in the prophase. So I will not be making cell membrane. Okay, I will be directly showing you with the nuclear membrane. Now in the prophase, what happens? This double layer nuclear membrane, right now it is continuous. Now what will happen? This nuclear membrane in prophase, it will starts to disintegrate, or it will starts to disappear. Okay, and the nucleolus that is present over here, it will also disappear. That means in the prophase there is a disappearance of what? Nuclear membrane, and this nucleolus that was present, it will also disappear. Okay, so I'll make it disappear. Okay, so it disappear. Now this chromatin fiber, that was the thread like structure, this chromatin fiber will be condensing. Okay, it will be condensing to form what? Chromosomes. Okay, it will be forming what? Chromosomes. And right now these chromosomes have how many chromatid? Each chromosome is carrying what? 2 chromatids, right? The diagram will be somewhat like this. That means one centromere, one chromosome, and one chromosome is having how many chromatids? 2 chromatids. Okay, so here I will be taking example of 2 chromosomes. Okay, now see what is happening. These chromosomes, this is one chromosome, this is again the second chromosome. Okay, like this, how many total chromosomes will be there? 46 chromosomes will be there. How many? 46. And these 46 chromosome, okay, they are not chromatin fiber anymore in the prophase. They will be chromosome, and each chromosome will be having 2 chromatids. And total how many chromosome will be there? 46. And all these 46 chromosomes will be scattered inside the nucleoplasm. That means inside the nucleus. Okay, they will be scattered. Got it? Now, as we know that in the interphase, the centriole also divides. So what will happen? The centrosome, right? The centrosome consists of pair of centriole. So the centriole starts to move at the opposite direction. So that means one centriole will be at the separate pole and another will be at opposite poles. That means centriole has moved to the opposite ends, and these chromosomes are scattered inside the nucleus, that is in the nucleoplasm. So this is what happens in the prophase. Now the second one comes the metaphase. Now what actually happens in metaphase? These chromosomes that you see, so I am taking the example of 2 chromosomes. So what happens? The nuclear membrane has already disappeared. Now I will be showing you here the cell membrane. Okay, nuclear membrane has already disappeared, so I'll not be making it properly. Now what happens? These chromosomes will be arranged like the equator, right? You know, equator, the line passes on the globe, I guess, right? In geography you might have studied this is the earth, and the equator is the line that passes from India also South Africa. Okay, I'm not good at geography, so don't take this advice from me, but I think you are understanding what I mean to say. That means in a single line, these chromosomes, right now how many are there? 2, but actually in each cell how many are there? 46. So these 46 chromosomes will be aligned in a single line, okay, one by the other, okay, next to each other like this, and the chromosome will be aligned. So in metaphase, what exactly is happening? The chromosomes are aligning and they will be forming a single line, and this actually the structure that will be forming, we will be calling it as equatorial plane. Okay, we will be calling as equatorial plane, or we will be calling it as metaphasic plate. Okay, because it is straight away flat structure, so we will be calling metaphasic plate. So equatorial plane or metaphasic plate. Just we have to remember in metaphase that it will be a single line arranged like the equator, and we have the centrioles over here, we know this. Now what will be happening? From the centrioles there will be the origination of the fibers. Okay, there will be the originations of the fibers, and these fibers are called as spindle fibers. Okay, in your Selena book they have written one word, achromatic spindle fibers. Okay, achromatic over here means that these spindle fiber will not be taking any stain if stained for the laboratory purpose. Okay, they will not be visible under the microscope properly if they are stained. So that's why we are giving the term achromatic. But you don't have to worry about this. You just have to remember that these are the spindle fibers. So these spindle fibers are arising from the centrosome from the both the direction. Okay, so this will be happening in what? Metaphase. Now the next one we have is the Anaphase. Okay, stay with me. Okay, don't go out of focus. This is very important. This is the basic thing that I am telling, and don't worry, we will be discussing from the book also step by step. Just this is the concept that you have to understand. Now see, after the arrangement of the chromosome in the metaphasic plates, what is happening? So that means the next phase that will be coming is the Anaphase, Prime Minister at, okay, this A. Now in Anaphase what will be happening? Okay, this is centromere. Okay, I'm trying to make good diagrams so that you can understand properly. Okay, I have to remove this one. Okay, I have removed. So let me draw it a little bit away. This is the chromatid. Okay, so like this, how many have already arranged? 46. And right now I am showing you how many. 2. Okay, I'm showing how many. 2. Now the spindle fiber, this is the centrosome, this is the centrosome, and each centrosome is having 2 centrioles, that is the pair of centriole. Now what will be happening? The spindle fiber, it will be attaching to the centromere by a special structure that is present on the centromere, and this special structure is called kinetochore. I have already told you in the very first lecture. Okay, so this spindle fiber originating from the centrosome, it will be attaching to the kinetochore, and in the same way this centriole will be attaching, will be sending the spindle fibers, and they will be attaching to the kinetochore. Now in the anaphase, what will be happening? These spindle fibers, they will begin to contract towards the opposite ends. This spindle fiber will be contracting this side, and this spindle fiber to the opposite side, because of which what will happen, here the pull force will be coming, and here the pull force will be in the opposite direction, because of which what will be happening, the centromere will split into 2. And now because the centromere will be splitting into 2, what exactly we will be getting? Let's see. Okay, so what we will be getting, this is the centriole, this is the centriole, and now this is the separated centromere, and these are the chromatids, and these are pulling this side. This is the centromere, these are the chromatids, and this is pulling on the same side. This is the centromere, these are the chromatids, and they are pulling in this direction. This is the centromere, these are the chromatids, and they are pulling in this direction. So you can compare the diagram right now. Over here, this whole thing will be splitted into half. See, right now how many chromosomes are there? 46, and 46 chromosomes are there, but how many chromatids are there right now? 92. And according to this diagram, how many chromosomes? 2 chromosomes and 4 chromatids. Now, and this is happening in the same cell, in the same nucleoplasm, right? Now over here, what will be happening? See, in the very starting I have told you, the number of chromosome depends on the number of centromere. Now total how many centromere you can see over here? How many? 4. That means 4 centromere are there. That means 2 centromere in this direction, 2 centromere in this direction. This means 2 chromosomes over here and 2 chromosomes over here. In the same way, 2 chromosomes are there, and how many chromatids? 2 chromatids. Okay, don't get confused. I hope you're not getting confused. That's why 2 chromatids are there. Let me explain you again. This is what? 1 chromosome with 1 chromatid. So 2 chromosome with how many chromatids? 2 chromatids. I hope you got it. And in the same way, 2 chromosome and 2 chromatids. Now see, we have started from where? 2 chromosomes, right? And how many we are getting? 2 chromosomes, right? We are having this one is separated and this one is separated, but this is happening right now in a same cell. The division of nucleus is taking place right now. So how many chromosomes are there? 1, 2, 3, 4. How many chromosomes? 4 chromosomes in total. And how many chromatids? 4 chromatids in total. Now see what specific happens in this anaphase. This chromatid that is separated, we will be calling this same chromatid as chromosome. I hope you are not getting confused, and if you are getting confused, don't worry, we will be discussing from the book also, then you will definitely understand. So what's happening over here? This part is separating this side, and this part is separating this side. Now let me explain this more properly. We were having how many? 46 chromosomes and 92 chromatids. Now what is happening? Out of 92 chromatids, 42 chromatids are going this direction, 46 chromatids are going in this direction and 46 chromatids are going in this direction, in the opposite direction. And these chromatids are now called as what? Chromosomes. They are called what now? Chromosomes. Okay, so right now the cytoplasm has not yet divided, only the nucleus is dividing. So over here, what is happening? The separation of the chromatid is taking place in the opposite poles as per the centrosome. So this was about what? The Anaphase. Okay, now next comes the telophase. Let's begin with the telophase. Okay, things are a little bit complicated, so I need you to have proper focus, else you will not be understanding. Okay, and if you are not able to understand, so you can leave the comment down below. I'll try to make it clear once more by the comments. Okay, now see, this was the cell, right? Here is the centriole, here is the centriole. Half of the chromosomes, they have reached here, and we were discussing with 2 chromosomes, and half of the chromosomes have reached here. We were discussing about 2, so 2 are over here, 2 are over here. In the same way, if we take the example of 46 chromosomes, 46 are over here and 46 are over here. But together, how many chromatids are there? There are total how many chromatids? 92 chromatids. Okay, 46 chromatids over here. See, these are the chromatids, these are the chromosomes. Both are same. Now in the telophase, what's happening? They will be moving separately, and the nuclear membrane and the nucleolus that was disappeared, it will start to reform again. Okay, it will start to reform again. The centromere is actually outside the nuclear membrane. Okay, so the nuclear membrane will be forming, the nucleolus again will be forming. So nucleus is divided, but the cytoplasm is still not divided. So this is what happens in the telophase. Okay, so we have completed what? Karyokinesis. We have completed what? Karyokinesis, and in the karyokinesis we have discussed prophase, metaphase, anaphase and telophase. After this, the nucleus is already divided, and the next step now will be cytokinesis. Got it? Now let's switch to the book so that you can understand more properly. So this is the content from Concise Serena book. So let's understand with the help of the diagram. Okay, the pen is working. See, after the interphase, we will be having what? Prophase, A and B. Okay, this A and B means this is the A diagram, this is the B diagram. Let's see the interphase. In the interphase, see, cytoplasm is there, centrosome is there, nucleolus, nuclear membrane, and right now they are in the form of chromatin threads. Okay, in the same way it's in the plant cell also, and cell wall is made up of cellulose. You already knew it. Okay, everything is same. Now what will happen? After interphase, what will begin? Prophase. Now in prophase, you can see very clearly that these centriole, they have started to move in the opposite direction, and after moving they will be reaching in the opposite direction. And now these chromatin fiber, they have been condensed and they took the shape of the chromosomes. Okay, in this diagram they have shown how many? 1, 2, 3 and 4. So they have took the example of 4 chromosomes to make you understand. In the same way, in the plants also they have 4 chromosomes. Now what you have to remember is, in the plant there is no centriole, right? There is what? There is no centriole. So, and we know that centriole helps in the initiation of cell division. So who will be dividing the cell over here? We will be discussing it later. Don't worry, it's a very good point. Okay, an important point, but not in the syllabus, but still we will be discussing. Okay, so we'll see over here in the early prophase, if you can see the centriole, okay, the centrosome, they have started to move in the opposite direction, and in the late prophase they have moved in the opposite directions, and these are the 4 chromosome, those are scattered. In the same way here it is scattered. See, centriole or the centrosome helps in the formation of the spindle fiber. The spindle fiber that moves to the opposite centriole or towards the opposite centriole, these are called the achromatic spindle fiber. There are some fiber that are in the opposite direction. These are called as ester rays. Okay, and these ester rays are arising from where? The centriole. Now over here there is no centriole, but spindle fiber is there. Because of the absence of centriole, there is no ester rays. That means ester rays are absent in plant cell. Okay, this will be the difference. So here is the spindle fiber. Now in the metaphase, you can always read this content. Okay, you can pause the video and you can read what is written over here. Okay, so let's begin with the metaphase. Now in the metaphase, what's happening? These 4 chromosomes are aligning. See, the centrioles are in the opposite direction and these chromosomes are aligning in a straight line. Chromosomes lie along the equatorial plane of the spindle, in the same way in the plants, in the same way in the animal cell also. Now the next phase is the anaphase. In anaphase, what is happening? Centromere attaching the 2 chromatids splits or divides. So just compare the diagram properly. See the diagram over here. These were the 4 chromosomes. The centromere is splitted out and they have been separated. Now these are the 4 chromosomes, these are the 4 chromosomes, these are the 4 chromatids, these are the 4 chromatids. Okay, these will be starts to move in the opposite direction, and after moving in the late anaphase, they will be reaching over here, these will be reaching over here, and this is how the anaphase will be completed, as these chromatids have reached in the opposite direction. Now the very important point over here, I guess it's not written over here, they should have written that these chromatids which have been separated are now termed as chromosome. This is very important line, and every student gets confused that if the chromosome is separating, they should be half. Why they are the same? Why? Because we are calling these chromatids as chromosomes only. Why? Because the number of chromosomes decides who? Decided by the centromere. So 4 centromere, 4 chromosome, right? 4 centromere, 4 chromosome, but they have been splitted out. Okay, so next we will be discussing about telophase. Now see, in the telophase, what's happening? The nuclear membrane has reappeared, the nucleolus reappear over here. They have not shown the nucleolus here. Yes, this is the nucleolus, and the nuclear membrane reappear, and these chromosomes, they again decondensed, they have again formed what? Chromatin fibers, from where we have started, right? From the interphase, again they have taken the shape of what? Chromatin fiber. But right now only the nucleus has been divided. The cytoplasm is still left to divide. This whole theory part you can read, pause the video and read everything. Right now we will be discussing the difference between animal and plant cell for the mitosis later. Now what will happen? The last thing after karyokinesis, what will begin? Cytokinesis. And in the cytokinesis, what will happen? The formation of furrow will start. Okay, furrow formation is taking place, and over here the formation of cell plate is taking. We will be discussing in detail just after 1-2 minutes. So after furrow formation, what's happening? We have now 2 daughter cells. So from a single cell with a single nucleus, we are having how many? 2 daughter cells, and the number of chromosome is still maintained. I hope you understood. Okay, so you can pause the video, read it out. You can pause the video and read this content. So the basic thing about mitosis is already been completed. Let's discuss the differences written here, and we will be switching to Sarita Garwal book also for the differences. They have explained very properly along with the proper diagram also. In animals, esters are there, and because centriole is absent, ester will not be formed. Cytokinesis happens by furrow formation, and in cytokinesis for the plant happen by cell plate formation. in most of the tissues of the body, the meristematic tissue in for the plant, meristematic tissue will be there for lengthening and the increase of girth. And in the animals, the somatic cells, in the very first lecture I told you, the somatic cells will be taking place for the mitosis, right? Means mitosis will be taking place in the somatic cells. Let's switch to the Sarita Agarwal and let's see what's written over there. This is the content from Sarita Agarwal book. I will not be discussing everything. You can pause the video, take the screenshot or read. Okay, so this is Prophase, Metaphase, this is Anaphase and Telophase, everything properly written over here along with the beautiful diagrams. Okay, you can take the help from here. And then we have the cytokinesis. You can take the screenshot right now. This is continued, the cytokinesis. You can read this part. Okay, now let's try to understand with the diagram. In cytokinesis, what's happening over here? The furrow formation is taking place, and in here what is happening? Cell plate formation is taking place. Now see what's happening. This was the cell membrane. The cell membrane is invaginating inside. It is going where? Inside. So the depression is forming here. That means furrow formation, it is taking centripetally. Okay, the centripetally and centrifugally words you will be understanding in physics, in circular motion or angular motion, rotational motion, whatever it is. But right now just understand, suppose this is your hand and you have tied a stone with the string, and this is the stone, and you are moving this stone in a circular direction. So from your hand towards the periphery, the force, centrifugal force will be taking place, and from periphery towards the center, centripetal force. So over here if you can see what's happening, from the periphery towards the center, the force is taking place over here, due to which the furrow formation is taking place. That means the force is centripetally. In cell plate, what is happening? From the center, the cell plate is moving towards the periphery, so it is centrifugally. I hope you understood. Rest you will be understanding in physics. Okay, so after the furrow formation is completed, the two daughter cells have been divided and they are separated. So nucleus is also separated, cytoplasm is also separated. In the same way, the cell membrane is separated, and we have two separate daughter plant cells. Okay, and the differences also we are having. Centriole is present, centriole is absent, centriole is absent, so esterase is also absent. Here esterase is present. No cell plate formation, but furrow formation takes place. Over here cell plate formation takes place, but furrow formation is absent, and this happens in the somatic tissues and this happens in the meristematic tissue. Okay, so the basic points, everything is completed. Now here are some of my questions. Now if in the plant cell centriole is absent, so how is the initiation of cell the cell division is taking place, how the anaphase is taking place? See, in the centriole we have seen that spindle fiber occurs, formation takes place, and in the opposite direction there are ester rays. In the plants the centriole is absent, but still there is a formation of spindle fibers. And how this spindle fiber formation takes place, this is not yet been discovered. Yes, the scientists are still working on this, and let me show you one image so that you can be sure that what the research is still going on. Look at this image. This image is from one of the journal from the scientist, and this has been published by NCBI, National Center for Biotechnology Information. Okay, so I want you to focus on this line where it is written. See, plant cells lack such structured microtubule organizing center, and some of their microtubules appear to nucleate from near the nuclear envelope. But now here is the line, but very little is known about spindle formation in plants. Understood? So these three lines are important. Very little is known about spindle formation in plants. So still we don't know how these spindle formation takes place, but the formation takes place with the absence of centriole, and the plant cell also divides. Okay, if you want to take the screenshot of this also, you can pause the video, take the screenshot. This is the comparison from Sarita Agarwal book for mitosis in plant cell. Okay, A, B, C, D, E, and we have late telophase. Here the significance are written. So this was it, and now let's come to the very important calculation. Now you see the chart over here. First of all, pause the video, take the screenshot, save it in your this gallery or whatever, so that you can take the help again and again whenever you get confused. Okay, so let's discuss. See, in the prophase there are 46 chromosomes, but because in the interphase S phase they have already been replicated, so there were 46 chromosomes but 92 chromatids. Now in the metaphase, again 46 chromosome, 92 chromatids. Now see what exactly happened in Anaphase. The centromere had splitted out, and because of the split of the centromere, 92 chromosomes have been formed. Okay, 92 chromosomes and still 92 chromatids. In the telophase, because it is a nuclear division, cytoplasm has not yet divided, so same 92 chromosomes and 92 chromatids. But as soon as the end of mitosis, the cytokinesis took place, the cytoplasm divided, these 92 chromosomes divided into separate cells. Now each cell, separated cell, is having how many chromosomes? 46, and how many chromatids? 46. So make sure to remember these numbers, that how the chromosomes are formed properly in the correct sequence and numbers. Okay, this is not in your syllabus, so don't worry, this is about meiosis, but basic things are there which we will be discussing in the next lecture, and the next lecture will be the last lecture. And I think this video is a little bit long, but if you have watched this video very properly, this video will be worth it. And this video, this third lecture, okay, this will be the very important lecture of this whole this chapter, cell division, and you can look at this lecture again and again if you don't understood cell division. I have tried my best to explain cell division very properly, step by step, so rest is upon you only. Make sure to learn it properly. And so this was it, and next lecture will be also uploaded soon, maybe tomorrow or day after tomorrow. So stay safe, stay at home, enjoy your lockdown period, and keep studying and be motivated. Watch movies also, but don't forget to study. So wish you good luck, keep studying, keep rocking, and make sure that you watch Amoeba Classes, because for biology, you know Amoeba Classes is the best. Bye bye.